Thank you.
Oui.
Je vais... Ah non, c'est bon, normalement le focus est mis, donc les mails ne devraient pas arriver. Ça va.
Yes, we see you, Marke. Okay, hello. Welcome to this second Discover Glow 2 webinar with a focus today on nanolux technologies. So, and more specifically, we will focus on two topics, the nanobit complementation reporter assay and Lumit immunoassays. We will have a talk of Dr. Julien Ronson, and then we will follow with a short Q&A session, and then we will proceed with the presentation from Dr. Dan Lazar, again with a short Q&A session. I would first like to add some practical points. So please feel free to add your questions to the Q&A box. And please also add your name, so we are really sure to give back your feedback to you personally during the separate Q&A sessions or afterwards. We will also make a recording of this webinar available for you, and the slides from Dan can also be made available for you afterwards. The slides for Julien will be, you can reach out to him then directly if you have further questions. So let's move to the first talk from Dr. Julien Ronson. He's working at the University of Liège in the laboratory of molecular pharmacology, and he is working as a principal investigator. His research is focused on G-protein coupled receptors, actually notably on orphans. So yeah, Julien, be my guest and enjoy the webinar. Sorry, Julien, you have to unmute yourself. We can't hear you.
Oh.
Yeah, that's working. Thank you, Vince.
You're welcome. Sorry for the technical problem. Yeah, so hi again.
Could you maybe start again with your introduction? We missed you. Thank you.
Yeah, so I was just saying hi to everybody, and thanks for the organizer for Promega to give me the opportunity to present my work. That was made possible with some of the Promega technologies that I will be happy to explain to the audience. So the topic of today will be on the detection between G-protein coupled receptors and G-protein interactions. So what are GPCRs? G-protein coupled receptors are membrane receptors that have 7 transmembrane domains, and they have the particularity to couple to so-called G proteins that are composed of 3 subunits, alpha, beta, and gamma. And actually, they are really important receptors because we have a lot of them, so 350 non-odorant members, and they are key players in numerous physiological processes like vision, neurotransmission, metabolism, or endocrine regulation. And actually, another particularity of the family is that many of them are orphans, meaning they lack active or known ligands. and an important number of them are understudied. So why are we studying those receptors? Well, they are really well involved in physiology, but even maybe more importantly, they are currently the target of 30% of the marketed drugs, and this proportion increased to 60% if we consider also drugs acting indirectly through a GPCR. So several important drug families target these receptors, like molecules against pain, like opioids, drugs against asthma, like beta-2 mimetics, depression, allergy, and psychosis, the neuroleptics being D2 blockers, and we will come back to this later. So actually, only a few members of the family are exploited in these therapeutical areas, meaning that the potential for new drugs on those receptors is considered as really huge. So a few words on GPR signaling for those of you that are not familiar with this. So actually, we have this trimeric G protein inside the cell, and when the ligands that can be of different type, you see the ligands for GPCR are really various. When the ligand binds, then you have dissociation of the G protein between a G alpha subunit that exchanges a GTP for a GTP and the beta gamma subunit. And the receptor is then desensitized by phosphorylation of its intracellular parts, and a scaffold protein called arrestin binds to the receptors and brings it, in most cases, inside the cell. So these main pathways of GPCR can mediate most of their actions, and depending on the subtypes and the family of the G proteins, alpha subunits, like you have the main three families, alpha I, alpha S, and GQ, you can see that they are coupled to different cellular events mediated through adenylate cyclase for modulation of cyclic AMP second messenger levels, or through PLC beta to modulate calcium levels. But the beta gamma subunit, which is an heterodimer, can also activate its own pathways, and inside the cell through the arrestin component, it mediates desensitization, internalization, and there are also many evidence of non-clinical signaling that is currently the topic of important research to see how this arrestin could play a role in GPCR signaling. So, so here it's a summary of what can be done with these receptors, and you see that actually mostly they can be divided in two main events: the G protein coupled components and the arrestin component. So to really get to the points of the topic of today, I would like to bring another level of complexity in the signaling, which is the biased signaling. Well, it is a concept that has been proposed many years ago and that is now well described. And this concept tells us that actually, depending on the ligand that is bound to the receptor can adopt different kind of conformations, and this conformation can couple to intracellular transducer in a different way. So for instance, you can have conformation 1 that activates G protein signaling and endocytosis through arrestin, but the conformation 2 that activates only G protein signaling. And one can see that because of this many transducer of G protein activation, the possibilities in terms of bias signaling is it has huge potential, and it can also lead to better drugs or to advanced therapies. And one of the challenge, and it's the topic of the presentation, is to detect this bias, to be able to develop, to identify molecules that are biased towards some pathways and to develop new drugs. So this is not an easy task. I will try to explain why. So most of the studies actually try to, or so far, have identified bias between the G protein component and the arrestin component. But the bias for different G proteins has not been very well studied so far. So studies are based on different assays. So to be able to detect a bias, you need to be able to measure what is activated through the G protein pathway or through the arrestin pathway. And actually, technologically speaking, the assays, historically, the assays that measure G protein activation detect second messengers that are generated like calcium or cyclic AMP, and the assays that detect the arrestin activation through GPCR detect interaction between the arrestin and the GPCR, because the arrestin has a tendency to form a stable complex with the receptor and not to have an activity that is like G protein in generating second messenger. So it was logical to do this way. But actually, if you think in terms of pharmacology, it means that the G protein assays, most of them, so that are based on second messenger, detect the activity of a complex between the ligand and the receptor, whereas the assays that detect arrestin binding to the receptor measure a concentration of ligand receptor and arrestin complex, which is slightly different, and that leads to bias, but that are not dependent on signaling bias, but on system or optional bias, which means that you will see different response in your assays, but not because the ligand is biased, but because the assays are different in their principle, because you do not measure the same thing. Here you measure like an enzymatic activity, some generation of cyclic AMP or calcium, and here you measure an interaction. So one of the solutions for this for example, G protein interaction and not only its activation. So, so this as so to study the bias for G protein as an interest. Of course, so far it has been done, like I said, mostly between G protein and arrestin that are really different in the cell, but actually it has some also interest to detect the G protein bias. The problem is that we have a lot actually of G proteins. So 4 main families, so GI family, the GS family, the GQ family, and the G1213 family. And you see that these G
Sorry, apparently we have a little problem. Est-ce qu'on a toujours bien Internet? Oui. Bon.
PowerPoint.
Can you still hear us, Lindsay?
Yeah, we can see
Okay.
you. Yeah.
Sorry.
Yeah, there was a bit of yeah, it was yeah, not so going well. We heard about some internet issues, but I think it's working now.
Okay.
I do wonder if turning off the video feed might help.
Yeah, I would turn off your video for a moment. Just talking.
I will do that. I will stop sharing the video.
Yeah.
Okay.
Yeah.
All set.
Thank you.
Okay, so sorry for that. So yeah, so I presented this oversimplified scheme showing that the family of G protein was important for the downstream pathways, but actually it's much more complex than that, because each family has several members, but these different members bring to the same cellular event. So if you want to study the subtle regulation, it's kind of complicated by the fact that if you measure cyclic AMP, you will not distinguish which particular G protein would produce these changes. So if you want, for instance, to study bias between G alpha I and G alpha 2, and you use cyclic AMP, it's not possible to do it. So this is one of the problem. And the other problem is that actually most receptors couple to multiple G proteins families. So you can have receptors that are coupled both to GS and GQ. And if you want to measure the different contribution of each G protein to the activation of the receptor, using these downstream assays doesn't give a straightforward answer. So this can lead to difference in the receptor response depending on the tissue, because the different G proteins can be differentially expressed. It can also result in signaling in different cellular compartments, but it can also be used to bias the response of some receptors towards particular G proteins. So what can we do to measure this differential coupling to different G proteins? Well, the current strategies usually rely on resonant energy transfer technologies. And in these technologies, you use a donor that will emit some photons and an acceptor that is fused on the signaling partner. Here we would have the G protein. So when the G protein interacts with the receptor, you can detect the transfer of energy between the donor and the acceptor and measure at a particular wavelength the signal. So there are basically two modes for this: the bioluminescent, which is based on the luciferase fused with the receptor, and fluorescent energy transfer, where the donor is a fluorescent protein that need excitation to transfer its photon to the acceptor, which is always fluorescent. So these technologies have brought a lot of information on the mechanism for G protein coupling to the GPCRs and gave really great information, but they have some limitation. Actually, they have, even if this has been improved over the years, a low signal to noise ratio. The signals that are measured are usually of small amplitudes. They may require complex setup, in particular for threat. They can lack sensitivity or specificity in the way it is measured, and it needs important modification of the receptor that may impact its physiology, because usually these donors and acceptors are really large proteins. So all these limitations, of course, do not preclude the study of receptors G protein coupling, but they can be problematic for screening or to study an important number of molecules. So to really be able to study bias, these are some limitations if you want to study a lot of compounds, or to identify in a large library some compounds that could be biased for some G proteins. So what we did in the lab, so we wanted to develop some things that would be compatible with screening to identify easily or more easily ligands for understudied GPCRs, or to be able to study bias toward different G proteins on known receptors. And for this, we used this nanoluciferase, which was published by the Promega company in 2012. And actually, one of the main advantage of luciferase is that it is small, of course, but it is also brighter compared to what existed at the time. And this technology has seen important development in the recent years, because in 2016, Promega reported a complementation system that was a system where the nanoluciferase was inactive by removing a small part of it, and this could be used for a complementation assay. And in this initial article, actually, a very nice scientific report actually. It was explained that based on this wild-type fragment, the natural peptide that would complement just a strip of some amino acids from the initial nanoluciferase, this was optimized to give rise to two other small peptides, the sequence 86 and sequence 114. So one was with very low affinity to try to avoid to disturb the interaction between two different partners. And this would be later called the small bites, and a fragment with high affinity that could be used in other type of complementation assay to force an interaction between two partners, and this would later be called the high bite. So far, this wild-type fragment, the natural peptide, has not been reported in the subsequent development of the technology reported by Promega, and I will come back to that because we used it a little bit. Because for our particular system, we first try to use this system to be able to detect the interaction between a receptor and the G protein. So for this, we used what was recommended by Promega, which is a small bite, with the idea of detecting this G protein receptor interaction. And actually, we did it. So this is for the D2, dopamine D2 receptor, and we could detect with a fairly good signal the activation of the receptor by dopamine, the natural ligand. However, for some other receptor, it gave not so nice results. Actually, D2 was more like an exception. For the H3, for instance, we could see nothing, even if we knew that the system could work. So we had the idea to try the other complementing partners, and in particular the natural peptide, because actually in the field it was already some complementation assays between G proteins and receptor were already been attempts with other luciferase, and it always failed. And one of the explanation was that was proposed was that actually the interaction between receptors and the G protein was too transient to see complementation. So we reasoned that actually the small bites in this particular system could be actually not the best one. Why? Because it would not stabilize the interaction between the receptor and the G protein, and then we would be not able to measure it with a good signal. Whereas the natural peptide has some activity for the large bites, which is a complementing partner for the peptide, and could improve the signal. And because our goal was not to study the interaction, but to have an assay with the highest signal possible, we thought it could be a nice alternative strategy. So we also tried the high bite to see if this could also lead to a better signal. And actually, when we use this natural peptide, we could then detect the interaction of the G protein, in this case of the H3, it's GI, with a ligand, an agonist for the H3. And even more interestingly, the high bites then gave not as good results compared to the natural peptide. So we reasoned that actually this natural peptide, this unmodified peptide for the nanoluciferase in the nanobit system could be the best choice for the interaction, to detect the interaction between G PCRs and G proteins. So what is important in complementation assays is to detect or to assess the reversibility of the system. It's really important to be able to not disturb too much the natural interaction. And logically, with the small bites, when we add an antagonist, it could disrupt the interaction, and you see that the signal goes back to basal. With the natural peptide, the dynamic was even better, so the signal was better, the interaction was more rapid, and the dissociation was not affected by the slightly increased activity or affinity of the natural peptide for the large bytes. Whereas for the high bytes, you could clearly see that in addition to reduced signal, less advantageous dynamic range, the dissociation was slower and did not reach the basal level, which confirmed the fact that the high bytes disrupt a little bit the natural interaction. So we confirmed also this dissociation behavior, this rapid kinetic with another receptor, the H3. So here it was the D2, and here it's the H3, another receptor coupled to GI. So here it's the agonist and here the antagonist, and you can see that if you measure over time, you see rapid dissociation of the complex. So with this in hand, we decided to expand the system to other G proteins with other receptors coupled to these other G proteins. For the beta-2 coupled to G alpha S, we see a nice increase in the signal. The H1 is coupled to the GQ family. And for both G alpha Q and G alpha 11, you have an increase in the signal, and the thromboxane receptor that is known to be coupled to G alpha 12 and 13 would also give a nice signal. So because we wanted to make this system able to study subtle regulation made by ligands, we benefited from the fact that the D2 receptor, which was our model receptor, is actually a really important drug target. It is a target for agonists. It is a target for drugs against Parkinson's disease. Agonists are used to relieve symptoms for Parkinson's disease, and antagonists are used in psychosis and also in emesis. So it's a very important target, and actually, because it's a receptor that has been really well studied, in this particular case, it is known that the ligand can show bias toward different members of the GIO families. And it is of high relevance because these G proteins that are inside the brain are not coupled exactly to the same thing. So in principle, in theory, the therapeutic profile of agonist of D2 could be influenced by their relative bias toward different members of the GIO family. So we did with dopamine a profiling, which was the initial idea to be able to do profiling toward all the G proteins. And you see that with all the construct we generated, this recapitulated well the known profile of the D2 receptor known to be coupled to GIO family. And you can see already here that the intensity of the signal depends on varies with different G proteins. So this is at a single concentration of dopamine. And of course, what was interesting for us was to have to see the bias, and for this you need full concentration response curves. So we did it for both G alpha I1 and GO that we selected as a proof of concept, and we could see that in the kinetic mode, the response of the 2G protein was slightly different, but they could be both responsive to antagonists. And what we did also is that we created stable cell line and could determine what is called the Z prime factor, which is an indicator of the goodness of the assay in screening mode. And we did screen a small library of known compounds, and we could find known D2 agonists in this library. So the system was able to detect these dopaminergic agonists in the library. And we did full concentration response curve toward G alpha I, G alpha O for all the agonists that we found in our library. And you can see that actually the response is slightly different if you compare G alpha I and G alpha O. These agonists do not display the same EC50 and Emax depending on the pathway. And if we do this analysis of the bias, so you compare the PEC50 toward G alpha O and G alpha I, and the Emax of the two pathways, and you do this, what is called bias factor, this equation, you put the numbers, and we could see that ropinirole, which is a D2 agonist, was biased toward G alpha I1, and apomorphine, another D2 agonist, had a tendency to activate more the G alpha O protein. So in conclusion, for this assay, what we want to do at Future Prospect is to try to expand this to other receptors, to validate the observed bias on dose-limiting pathway to see if it can have an impact on the cellular physiology or the organ physiology, to try to see if this can be linked to a therapeutic efficacy or side effects, but importantly, to identify new biased ligands with these assays based on G protein. The system has been also described with mini G proteins, so other people have reached similar results with the nanobit system, and to also expand it to arrestin, with the idea of being able to study both arrestin and G protein together, and these are the results with beta arrestin, showing that it works also the system with beta arrestin. So I would like to thank you for your attention and to acknowledge people from my lab, and in particular Céline Lachet and Nadine Dupuis that performed the experiments. And I would like to stress that the plasmids are available at Gene. If you want to implement the assay in your lab, it's open and shared, and you can contact me further if you have any questions. So thank you again.
Thank you, Julien.
Maybe we can turn the camera back on.
Yeah,
yes.
Or we can even actually move to the second presentation because of sake of time. Then we will take the questions together after the two presentations. So thank you very much, Julien, for your presentation. And now we will move to our second speaker, who is Dr. Dan Lazare. He is actually working as a group leader and senior research scientist at Promega, and he will present about a newer NUMET immunoassay technology. So Dan, the floor is all yours.
All right, thank you, Lindsay. I appreciate the opportunity today to share Promega's LUMET technology for homogeneous bioluminescent immunoassays. We've just heard a really nice presentation by Dr. Hassan showing the application of nanobit technology. I will give an introduction on LUMET immunoassay technology. And then after that,
sorry to interrupt then. Can
Sure.
you share your screen? Because we don't see your presentation.
You know, I did share. Perhaps someone on your end needs to activate my sharing.
That's strange because I don't see it.
Okay, let's see again. Maybe it dropped.
I think it's lost somewhere. Yeah, now your desktop is coming up again. Perfect.
Excellent. Okay, sorry about that.
Yeah, and then maybe put it into presentation
just move the box out of my way so I can put it into presentation mode.
Great.
You see the whole thing now?
Yes, perfect. Thank you.
All right. Again, I'm happy to have the opportunity to present on our LUMET technology for homogeneous bioluminescent immunoassays. I'm going to begin with an introduction to LUMET immunoassay technology, and then I'll provide 3 examples of ready-to-use assay content currently available from Promega. At the same time, I'll provide a description of how investigators can build their own novel LUMET immunoassays. Now, Dr. Hassan just presented a beautiful presentation demonstrating the application of NanoLuc binary technology. So the only thing I want to emphasize on this slide is that for LUMET technology, we're using large bit and small bit peptide, which have extremely low affinity for one another. So therefore, there's negligible spontaneous association and luciferase formation between those 2 subunits. In fact, Lumit immunoassay technology leverages nanobit technology to deliver a bioluminescent immunodetection platform. In its simplest configuration, primary antibodies to a target are labeled with large bit and small bit subunits respectively. When those labeled antibodies bind to their shared target, they bring the small bit and large bit in proximity to one another, forming the luciferase activity and generating luminescent signal proportional to the level of analyte in the sample. Some of the attractive features of Lumit immunoassays are that they deliver simple add and read formats that can be performed directly on cells and medium. They're fast, taking only 30 minutes to 2 hours to complete. They deliver excellent sensitivity in broad linear ranges, which significantly mitigates the need for sample dilutions. They use only a standard plate reading luminometer to acquire assay signal, and they're amenable to high throughput applications. Importantly, the Lumit platform includes ready-to-use kits for specific targets, as well as reagents to enable investigators to create their own novel detection assays. I'll be describing 3 different configurations of Lumit immunoassays. The first is the direct configuration, which uses labeled primary antibodies. The advantage here is it maximizes the number of antibody pairs that can be tested for assay development. In the second configuration, the indirect configuration, unlabeled primary antibodies are used together with labeled secondary antibodies. The advantage here is it enables rapid assay development. And the last configuration that I'll describe is a competitive binding displacement format, which I'll elaborate on in more detail later in the presentation. The first application I'll describe uses the direct configuration, and that is the Lumit cytokine immunoassays, which are no transfer, no wash, add and read assays for detecting cytokines released from cells in culture. Now, there are a number of reasons to develop cytokine assays, but the real opportunity we felt here was that with Lumit technology, we could deliver a faster, easier method to measure cytokines without a requirement for specialized instrumentation. As I mentioned, the Lumit cytokine assays use nanobit labeled primary antibodies, which have negligible interaction in the absence of analyte. So together with the inherent low background of luminescent assays, this provides for a very low assay background. However, in the presence of target cytokine in sample, the labeled antibodies converge on the target, forming the bright luciferase by the proximal large bit and small bit subunits, and generating a luminescent signal, again, proportional to the level of analyte in the sample. The workflow for the Lumit cytokine assays is very straightforward and completely homogeneous. Cells are plated and treated in a microplate format. To these cell wells, one adds the labeled antibodies and incubates for 60 minutes. Subsequently, you add the Lumit detection reagent, which contains the luciferase substrate, and just a few short minutes later, you can record luminescence with a total assay time of about 70 minutes. Although I would hasten to add that the signal half-life is about 2 hours. So because of the glow kinetics, this makes the assay very amenable to batch plate processing. On the right, we see example data from 5 different Lumit cytokine immunoassays. And what we're looking at is a log-log plot of background subtracted luminescence versus concentration of analyte. And the first thing to point out is that all of these assays have very broad linear ranges, actually well in excess of 3 logs of cytokine concentration. In addition, they deliver excellent sensitivities with lower limits of detection, less than 10 picograms per mil. And in data not shown here, they all deliver high signal to background ratios. So at the top end of the calibration curve, the signal to backgrounds are typically 1,000-fold or more. So how do you build a direct Lumit immunoassay? Well, it starts with the selection of primary antibodies. Subsequently, those antibodies are labeled with both small bit and large bit. And then you perform a rapid screen to identify productive antibody combinations for detection of the analyte. And lastly, you take your lead antibody pair, or maybe a couple pairs you're most interested in, and you optimize the concentrations of the individual antibodies in the pair for that particular assay. So for selection of the primary antibodies, you need to begin with purified antibodies with specificity and high affinity for the target of interest. Monoclonal antibodies, of course, may provide excellent sensitivity and specificity, but monoclonal and polyclonal antibody combinations can also be effective. In fact, ELISA-validated antibody pairs are often a good starting point, but they're not necessarily going to deliver the best LUMET assay. Because we're using labeled primary antibodies, so there's no need for use of secondary antibodies in the assay format, there's no restrictions on the species of primary antibodies that can be combined. What this means is, as we increase the number of candidate antibodies, it significantly increases the number of combinations available to test, and this leads to a nice improvement in the probability of technical success. To label the antibodies with small bit and large bit, we use the LUMET immunoassay labeling kit. And this allows for conjugation of the nanobits to the exposed primary amines on antibodies in 2 simple reaction steps. At the lower left, we start with purified antibody solution, and we add the amine reactive halotag ligand and incubate for 90 minutes. Subsequently, the reaction is buffer exchanged with Zeba columns to remove unreacted ligand, or we could use dialysis. This generates that primed antibody there in the middle of the lower part of the screen that has now affixed to its surface a number of halotag ligands. We can then incubate overnight with halotag large bit or halotag small bit fusion, which leads to the covalent conjugation of these subunits to the antibodies, leading to the production of antibody conjugated with large bit or antibody conjugated with small bit. Here we see an example of 7 monoclonal antibodies, arbitrarily labeled clones 1 through 7, for human interferon gamma. And these antibodies were labeled with small bit and large bit and then resolved on a gel. And we can see the small bit labeled antibodies on the left side of the gel and the large bit labeled antibodies on the right side of the gel. And what you can notice is a ladder effect. And this is because you have heterogeneous populations of labeled antibody with different numbers of subunits affixed to individual antibody molecules. What we're striving for is good representation in the areas of 3 to 5, perhaps 6 subunits per antibody molecule, and those are well represented in these samples here. Important note is it only took 2 days for the antibodies to be labeled and analyzed by gel. Once we have the labeled antibodies, we can perform a rapid screen for productive antibody combinations, and we do that by using a combinatorial matrix of the labeled antibodies in a 96-well format. Here we see these small bit labeled antibodies arrayed across the top of the plate such that they're dispensed down each of the columns, and the large bit labeled antibodies are arrayed on the side of the plate such that they are dispensed across the plate. This provides for a 7x7 matrix or 49 possible combinations of the antibodies that will be tested. The test is performed in 2 plates containing a submaximal concentration of the analyte to generate signal, and 2 plates without any analyte containing only culture medium to generate background. We can now take the signal and background data to generate signal to background ratios, and those results are represented here in a heat map. And you can see that there are a few combinations of antibodies that really light up and stand out from the other combinations. We can rank order the antibody pairs based on signal to background, keeping an eye on brightness and background. An important point here is it only takes 1 day to perform the rapid screen for productive antibody pairs. Now again, once you have a lead antibody pair or 2, you can now focus on optimizing the concentrations of the individual antibodies in the pair selected for use in the assay. Here's a simplified example of that, where we are keeping the concentration of antibody 1 and antibody 2 fixed in a 1-to-1 ratio, such that the x-axis concentration represents the concentration of each antibody in the pair. And on the y-axis, we're looking at a log plot of the luminescent signal. As we increase the antibody concentrations, you can see that the luminescent signal increases and tends to plateau at higher antibody concentrations. However, you can also see that as you increase the concentrations of the antibodies, you also increase the background signal, and it continues to increase at higher and higher antibody concentrations. So that if we now plot the signal to background ratio, we can see that a peak is achieved. And again, this is using submaximal analyte, but we still see that there is a peak signal to background ratio observed here at about 125 nanograms per mil of each of the antibodies. So this would be the concentration of antibodies we would select for further use of this assay system. I would point out that occasionally non-equimolar antibody concentrations can sometimes improve assay performance, and that can be evaluated by performing a more extensive combinatorial matrix of varying concentrations of antibody 1 against antibody 2. But in any event, it only takes one day to optimize the concentrations of antibodies selected for use in the assay. Now, once we have the antibodies, the workflow for application to a cell culture model is very simple. I've already described it earlier, but just a quick review here. The Lumet immunoassay workflow is completely homogeneous, very simple, and very fast, taking only about 70 minutes to complete. In comparison, if we look at the traditional ELISA workflow, it's certainly very heterogeneous, starting with the transfer sample from the cell treatment plate to a capture plate, but then having multiple steps of washes and media exchange and additions of reagents. And this is also a very time-consuming process, taking more than 5 hours to complete. So certainly the Lumet immunoassay workflows offers advantages of speed and simplicity. Here we see the application of Lumet IL-6 immunoassay for that rapid detection of cytokines released from human peripheral blood mononuclear cells treated in a 96-well plate. These cells are treated with 3 different stimuli: cell stimulation cocktail, which is a combination of ionomycin and PMA, lipopolysaccharide, and the TLR7/8 agonist R848. And the treatments were performed from 6 hours up to 72 hours. And what you can easily see is there are differences in either the magnitude or the kinetics of IL-6 release from the cells in response to these 3 stimuli. Importantly, all this data was generated with direct addition of the assay reagent to the cell wells with media present. There were no sample dilutions or wash steps involved, and again, it only took 70 minutes to complete the assay. Alternatively, if desired, sample can be transferred and one can perform split sample analysis for multiple cytokine determinations. In this case, the PBMC cells were treated for 24 hours, again in a 96-well plate, with the cell stimulation cocktail, LPS and R848. But in this case, the sample being transferred was split into 4 20 microliter aliquots, transferred into a 384-well assay plate, to which multiple Lumet cytokine assays were applied in order to generate these different cytokine profiles released in response to the various stimuli. Certainly, the simple Lumet cytokine assays allows for easy detection of cytokines released from more complex cell culture models. Here we're going to look at purified CD8 positive T cells or effector cells combined in culture with Raji-B target cells and treated with increasing concentrations of the bispecific T cell engager Blincyto. Blincyto binds to CD3 on the surface of the T cell and CD19 on the surface of the target Raji-B cell, leading to lysis of the target cells and concomitant cytokine release from the effector cells. Here we see the dose-dependent release of IL-2 detected with the Lumet IL-2 assay with a maximal signal-to-background ratio of 82-fold. And in parallel treated cell wells, we can see with the Lumet interferon gamma assay the dose-dependent release of that cytokine, now with the maximal signal-to-background ratio of 168-fold. And again, importantly, all of this data was generated with direct addition of assay reagents to the treated cell wells. So the Lumet cytokine immunoassays enable rapid add and redeterminations of cytokines released from cells, including quantitative assessments of T cell activation and differentiation. One point I don't have time to elaborate on today is that the 384-well performance of these assays indicates they're amenable to screening applications. There will be some data on that in the backup slides that'll be available when we distribute those slides. I do want you to know that current assays available for testing and purchase from Promega include IL-1 beta, IL-2, IL-6, interferon gamma, and TNF-alpha, and that other assays in development include IL-4, which is almost ready for external testing, as well as IL-10, VEGF, and others. And we're certainly very happy to receive recommendations for cytokines that we should prioritize for future development. So the next application of Lumet technology that I'll describe uses the indirect configuration with labeled secondaries combined with unlabeled primaries. This is the Lumet immunoassay cellular system, which is a lytic system for the interrogation of cellular signaling pathway regulation. We all know the crucial role that protein phosphorylation plays in cell signaling, and that specific phosphorylation events constitute important nodes in cell signaling pathways. The LUMET immunoassay cellular system provides a rapid, homogeneous assay system to quantify phosphorylation and degradation events related to key cell signaling nodes. The workflow for this LUMET assay system is also very straightforward and completely homogeneous. Cells, again, are plated and treated in a microplate format. To that, one adds the lytic reagent provided by Promega and incubates for 20 minutes to liberate the cell proteins into the lysate. You then add the combination of antibodies for detection of the specific target of interest and incubate for 90 minutes, followed by the addition of the LUMET detection reagent, and a few short minutes later you can record luminescence. And this assay system takes less than 2 hours to complete. In building a LUMET immunoassay cellular system, for example, for the NF-kappa B pathway, when we want to look at total and phosphorylated I-kappa B alpha, we'll need to select primary antibodies. So to detect total I-kappa B alpha, we'll use mouse anti-I-kappa B alpha combined with rabbit anti-I-kappa B alpha. However, to detect only the phosphorylated species of the protein, we'll again use the mouse antibody, but now combined with a rabbit anti-phospho I-kappa B alpha antibody. With both pairs of primary antibodies, we'll use LUMET secondary antibodies that are anti-mouse antibody labeled with large bit and anti-rabbit antibody labeled with small bit. Again, cells are treated and lysed, and then one adds the combination of primary and secondary antibodies for detecting the protein target of interest. In this case, the antibodies for detection of both phosphorylated and unphosphorylated I-kappa B alpha are used so that the luminescent signal generated reflects the total level of I-kappa B alpha in the sample lysate. However, if we use the specific antibody pair for detection of phosphorylated I-kappa B alpha, again with the same secondaries, the complete complex and luciferase formation only occurs now on the phosphorylated protein, such that luminescent signal here will represent the levels of phospho I-kappa B alpha in the sample. So this provides for rapid add and read formats to interrogate cell signaling pathways with no media removal, no transfers, and no wash steps. So how do you select the primary antibodies? Well, in this example, we're going to stick with the NF-kappa B pathway, looking at I-kappa B alpha as the key signaling node, using MCF7 cells treated with and without TNF alpha. We'll use labeled LUMET secondary antibodies and combine them with candidate primary antibodies. The antibodies, the primary antibodies, are used in a range of concentration of antibody 1 combined with antibody 2. And what we're looking at here is a heat map representing the fold induction of signal with TNF alpha treatment versus untreated cells. We can do the same thing with a second pair of candidate antibodies and a third pair of candidate antibodies, and it becomes pretty obvious that the first pair is the best pair. And we also know from this matrix approach the optimal concentration of each antibody in the pair to use for application to cells. We can then go further to validate the performance of the assay system, looking at linearity and sensitivity. In this first plot, we're looking at phosphorylated I-kappa B alpha levels now performed with TNF alpha treatment in the presence of the proteasome inhibitor MG132, where we can see the increase in luminescent signal relative to the increase in cell number. If we do the same thing with antibodies for a total I-kappa B alpha determination, if we look in the untreated cells now, we can see the increase in luminescent signal relative to the increase in cell number. In both cases, the data shows excellent linearity across a broad range of cell number and good sensitivity at low cell densities, working quite well down to as low as even 5,000 cells per 96 well. We can then go further to demonstrate the fidelity of the assay signal for the expected regulation of the NF-kappa B pathway. Here we're first looking at TNF alpha stimulated phosphorylation of I-kappa B alpha in the red bar. So we see the nice big increase in phosphorylated protein. But that if we co-treat with a pathway-specific inhibitor, IKK16, we can see, as expected, that response is blocked. Now down below, using the antibodies for detection of total I-kappa B alpha protein, we can see that the TNF alpha causes the expected decrease in I-kappa B alpha protein. But again, this response is blocked by the pathway-specific inhibitor. Now, the LUMET immunoassay cellular systems are not real-time kinetic assays, but because of the simple format and because with the addition of lytic reagent, which also contains phosphatase and protease inhibitors, all signaling is essentially frozen in their tracks, one can perform staggered treatment of various cell wells and then simultaneous addition of the lytic reagent to produce some really nice kinetic or temporal data. And here what we're looking at is the time-dependent increase in I-kappa B alpha phosphorylation by TNF-alpha treatment, peaking at around 10 minutes and then declining thereafter as the phosphorylated protein is degraded by proteasome. If we use the antibodies to look at total I-kappa B alpha, we can see that the TNF-alpha treatment causes a degradation of that protein that begins shortly after 5 minutes and then is markedly reduced by 30 minutes of TNF-alpha treatment. If the same experiment is performed in the presence of the proteasome inhibitor MG132, we can see the forced accumulation over time of the phosphorylated I-kappa B alpha in the lower plot, and then the greatly minimized reduction in total I-kappa B alpha due to the block of the proteasome activity. Now, the assay system is very useful for looking at drug modulation of the pathway. A little explanation of this plot: 100% represents the maximal degradation of I-kappa B alpha after TNF-alpha for, I believe, 30 minutes treatment in this case. So 100% is the maximal degradation. And what we see here is that the small molecule pathway inhibitor, IKK16, provides for a dose-dependent blockade of that degradation in both MCF7 cells and primary breast tumor epithelial cells. Likewise, on the right side here now, if we use antibodies which neutralize TNF-alpha, we can see these 3 therapeutic agents actually block I-kappa B alpha degradation. They block the NF-kappa B pathway activation. As expected, the potencies for these small and large molecules match what was predicted. Now, the LUMET assay is completely homogeneous, and if we compare its protocol to a couple other important and very useful quote-unquote homogeneous assay systems, I do want to point out a few differences. With the Surefire alpha lysa and the HTRF assays, one actually has to remove media prior to adding the lytic reagent, something which is not required for the LUMET assay. In addition, both of these assays have light-sensitive chemistry, so they have to be performed under alternative lighting. And as mentioned before, the LUMET assay takes less than 2 hours to complete, while the Surefire alpha lysa assay takes about 4 hours, and the HTRF assay more than 12 hours to execute. In addition, both the alpha lysa and the HTRF require specialized instrumentation for signal acquisition. Now, the LUMET immunoassay cellular system has been validated with multiple signaling targets, and a number of those are listed on this slide, and this has been performed in a great many different cell types, suggesting that this universal immunoassay system can be adapted for any pathway as long as there are appropriate primary antibodies available. In fact, the details of how to apply the assay system to these different targets have been worked out and are available in application notes with detailed technical guidance, including the identity and vendor source for specific antibodies to use for each target. And this information can be found on Promega's LUMET homepage. So one additional point I'll make as I wrap up on the LUMET immunoassay cellular system is that this system has also been demonstrated to work well in 384 well plates, which indicates it should be amenable to screening applications. So the LUMET secondary antibodies can be adapted to any pathway or pathway node of interest as long as there are suitable primary antibodies available from different host species. Promega makes available LUMET secondary antibodies for anti-mouse, anti-rabbit, and anti-goat, and all of these secondary antibodies are available with either small bit or large bit labeling, so they can be combined in any combination. And of course, also available are the lytic and detection reagents to allow you to apply the completely worked out assay systems that are available and described in the application notes, but also for you to build and develop your own novel LUMET immunoassay cellular systems. Now, the last application of LUMET immunoassay technology that I'll describe today uses a competitive binding displacement format, and that is the LUMET FcRn immunoassay, which is a solution-based add and read assay for antibody neonatal Fc receptor binding. Importantly, FcRn antibody binding can be modulated to increase or decrease the bioavailability and efficacy of antibody drugs. What was needed was a simple and rapid solution-based assay system to assess antibody F-serum binding to support therapeutic antibody development. To do this, non-specific generic IgG molecule was labeled with large bit to create a tracer, whereas the target F-serum was labeled with small bit, and when the tracer and target interact, they again form the luciferase and luminescent signal. However, in the presence of unlabeled test IgG, a competitive binding displacement scenario is set up so that when the test IgG binds to the target, it prevents the tracer from binding, thereby decreasing luminescent signal. And so the expectation is that as one increases the concentration of test IgG, you'll have lower and lower luminescent signal. And also as one looks at the binding isotherm, right and left shifts of the isotherm will indicate differences in affinity for the antibody for FcRn. The workflow again for this LUMET application is again completely homogeneous and very simple. Tracer, target, and test IgG are plated in microplate format and incubated for 30 minutes to allow for binding equilibrium to occur. You then add the LUMET detection substrate, and a few short minutes later, record luminescence. Again, in this case, taking only 30 to 60 minutes. And I do want to emphasize again, though, that the assay signal for all of these assays have glow kinetics with half-lives of 2 hours or more, again, allowing for batch plate processing. Here's some example data from 3 therapeutic antibodies applied in the LUMET FcRn immunoassay, and you can see that with increasing concentrations of each of these antibodies, you see decreasing luminescent signal. And if we calculate the IC50s, we see that they were very similar for all 3 of these antibodies as expected. They were expected to have very similar binding affinities for neonatal Fc receptor. However, if we take one of these antibodies, trastuzumab, and we incubate it with peroxide for increasing treatment times, this leads to the oxidation of a couple key methionine residues and decreased affinity of the antibody for FcRn, and that is reflected in the right shift in the binding isotherm with increasing treatment time. So this and other data demonstrates that the assay system delivers an excellent window for the study of antibody FcRn binding. And again, one other point I'll make here, in addition to the fact that the assay uses very low volumes of 10 to 20 microliters of sample, the assay format is also amenable to high throughput screening applications, and that data will also be available in the backup slides. And to summarize now on the platform, the LUMET immunoassay platform, it's a bioluminescent immunodetection platform based on nanobit technology to deliver rapid, no-wash, no-transfer assay formats with large dynamic ranges that can be performed directly on cells with media present. They require only standard plate reading luminometer for signal acquisition. They're all scalable for screening applications, and the LUMET platform includes both ready-to-use assay kits as well as reagents to create novel detection assays. And I do encourage you to, in addition to looking at some of the backup slides that'll be provided in the deck, to go to the LUMET homepage where you can find further information on the ready-to-use assay content, as well as the reagents that you can put in your toolbox to develop your own novel LUMET detection assays. So I do appreciate your time and attention today, and I'd be very happy to answer any questions you may have.
Thank you very much, Dan, for your nice overview presentation. Also, thank you very much, Julien, for both your presentations. And maybe we can move to some questions that were asked during the talks. I first ask some questions to you, Dan, about your talk. One question was, what is the order of sensitivity for Western blot, ELISA, and LUMET with respect to input concentration? Can you maybe give some feedback on that?
Well, one way to think about it is the fact that with the LUMET cellular immunoassay, you can work with as low as 5,000 cells in a well, and still typically with many of these assay systems get a very nice signal window. It'd be very difficult on a Western blot to be able to use as low as 5,000 cells and get a significant signal. I will add that a number of these assays were validated, though, or benchmarked against ELISAs to show similar fold or similar regulation of the phosphorylated proteins or the levels of the proteins. I could also add that in the backup slides, there's a comparison of the Lumit immunoassay cellular system for, I believe it's phospho-STAT 3, and it's a comparison of a Surefire alpha-LISA assay together with HTRF assay, and in that we show that the signal to background, or the sensitivity really for the Lumit assay, is as good or better than the alpha-LISA and much better than the HTRF assay, at least in that particular example.
Okay, great, thank you. Then another question, does the Lumit system also work for membrane-bound proteins, for instance, after transfection?
So if there's a couple scenarios to think about here. If you're talking about application to intact cells to look at surface exposure of membrane proteins, that is actually something we're working on and having success with. So you can use antibodies or other affinity reagents to cell surface epitopes and develop an assay in that way as well. If it's a membrane protein, also one, I suppose, could apply a lytic approach, although yeah, I mean, it depends on what your objective is. You could use a Lumit immunoassay cellular system with a lytic reagent, and then certainly it should work there as well.
Okay, thank you very much, Dan. Then I want to end up with some questions for Julien. Julien, there was a question about do endogenous G proteins affect bias measurements in your approach? Did you try G protein knockout cells?
That's a very good question. So it's something we try to see what could be the impact of endogenous G proteins, and they do have an impact because they can compete with the tools we use. But of course, in the system we implemented, we use large amounts of labeled G proteins. So the impacts of endogenous protein are minored. But of course, if one would want to study more the things in a more physiological context, then using cell lines that are knocked out for native G proteins would be a better system, of course.
Yeah, thank you. Then another one. It's just to clarify, but I think you partially already answered in this. So whether you have used nanobit tags expressed endogenously, so by knock-in or by overexpression. So
yeah, it was overexpressed, and of course, again, so a better system would be a knock-in, but you know, the idea being to get the best signal and the most intense signal possible. The idea is to put a lot of G protein. So if you want to make knock-ins, it's really to try to study the real situation, the real levels of G proteins. And for that, then one has to consider the fact that the complementation system, even if it's better than other ones, may disrupt a little bit the native interaction between the G protein and the receptor. So the conclusion you can make, yeah, you must be careful with this.
Yeah, I understand. Thank you. And then there's another question, and I will take this as a last question because we can't answer all the questions that came in. But another person is wondering whether you know a method to measure a decrease in cyclic AMP as a result of the activation of alpha-2 adrenergic receptor by an agonist, or is it only possible to measure an increase of cyclic AMP?
No, of course not. And I can promote another Promega product, which is a CMP glow sensor, which is terrific for measuring cyclic AMP in living cells in a kinetic way. We actually published another story, but it was 5 years ago, on the use of the CMP glow sensor for GI coupled receptors specifically.
Yeah, Certainly. So we will certainly come back to that person personally. So I really want to thank you, Julien, and also thank you, Dan, for your really great presentations and for your answers to these questions. I will have to send you extra questions because we can't answer them all because of sake of time. And I really want to thank everyone for its attention. We will make the recording available for you. Also, the slides from then will be made available for you. And I would advise everyone that has extra questions about a part of Julien to reach out to him personally. And whenever you have additional questions, please don't hesitate to contact us. And I just want to add, and also would like to invite you to our last session, which will take place on the 3rd of December, and it will be about cellular target engagement and protein degradation. So thanks again for joining, and stay all safe and well.
Thank you. Bye-bye.
Thank you. Bye.
Thank you.